everyone, today we are going to discuss about Chapter 2. Chapter 2 covers food microbiology, hygiene and sanitation. Before moving into the topic, let's see the syllabus. Food chemistry, food microbiology, hygiene and sanitation, food processing technology, food loss and organization, public and occupational health and nutrition. So, Chapter 1 has been discussed earlier and its link has been given in description and I button. Chapter 2 will be discussed today and remaining 3 topics will be discussed on upcoming classes. So, please subscribe our channel for more videos. Let's move on to topic. The term fermentation was coined by Louis Pasteur. And next about optimum temperature of psychrophil and mesophil. For psychrophil 10 to 15 degrees Celsius and for mesophil 30 to 45 degrees Celsius. And next, shapes of bacteria. Bacillus, rod-shaped. Coccus, spherical-shaped. Vibrio, comma-shaped. And next, black bread mold. Rhizopus stolonifer. Trichinosis, Trichinella spiralis in pork. Protozoa harmful illness by Entamoeba histolytica, Toxoplasma gondii, Balanchidium coli and Giardia lamblia. And next, flat source spoilage is caused by Bacillus coagulans and Bacillus stereothermophilus. Next, Thermophilus anaerobic spoilage is caused by Clostridium thermosacrolyticum. Sulfide spoilage is caused by Clostridium nigricans. Putrefactive anaerobic spoilage by Clostridium sporogens. Aerobic sporoformous spoilage by Bacillus species. Biological spoilage by mesophilic microorganism by Bacillus species, Clostridium species and yeast. And next, Campylobacter jejuni is present in poultry meat and unpasteurized milk. Listeria monocytogenes is found in milk, cheese, vegetables and meat products. Then Escherichia coli in water and cooked maize meat products. Next about rad-apatization, radicidation and radorization. So first, rad-apatization. It is equivalent to radiation sterilization. So the level of radiation here 30 to 40 kGy. Next, radicidation. It is equivalent to pasteurization of milk. So the level of radiation here 2.5 to 10 kGy. Next about radorization. It is considered as pasteurization. So the level of radiation is 0.75 to 2.5 kGy for fresh meat, poultry, seafood, fruits and vegetables and cereal grains. And next about pasteurization temperatures. 145 degree Fahrenheit, that is 63 degrees Celsius for 30 minutes and 161 degree Fahrenheit, that is 72 degrees Celsius for 15 seconds. Next about D value. It is the time required to destroy 90% of organisms. And next about Z value. Temperature required to reduce the population by 1 log cycle. And next about F value. Here it is time in minutes at 250 degree Fahrenheit with respect to its capacity to destroy the spores or vegetative cells. Then about controlled atmospheric storage. Here the product environment is maintained throughout the storage. And next, the composition changes during the storage as a result of product and microbial respiration. Next about nisin. It's the natural antimicrobial agent. It is produced by Lactococcus lactis. It is naturally present in milk. Next about intrinsic parameters. pH, moisture content, oxidation reduction potential, nutrient content, antimicrobial constituents and biological structures. And next about extrinsic parameters. Temperature of storage, relative humidity of environment, presence and concentration of gases in environment. And next, good hygienic principles, GHP. So here it is 8. Next, good agricultural practices, GAP, 4 pillars and 9 key elements. And next about good hygienic principles, water quality, air quality, drainage and waste disposal, temperature control, personal hygiene, lighting and storage. Next about good agricultural practices, pillars, economic viability, environmental sustainability, social acceptability and food safety and quality. Next about good manufacturing practices, 5 Ps. Here people, products, process, procedures and premises. Next about BAP, best aquaculture practices. Next, Fssc 22,000, food safety system certification. And bad Parker agar, it is for Staphylococcus enumeration, EMP for gram-negative bacteria isolation, NAMAS, National Measurement Accreditation Service. Here comes asepsis, keeping away from microbes. Next, removal of microbes by filtration, centrifugation, washing, trimming and blanching. Next, hindering the growth of microbes by refrigeration, drying and adding chemicals. And next, killing microbes by irradiation and heat processing. Next, the activities of foodborne microorganisms can be slowed down or stopped at the temperature above freezing and generally stopped at sub-freezing temperatures. Then chilling temperature. It is between the usual refrigerator temperature and room temperature, usually about 10 to 15 degrees Celsius. Next, freezer temperatures are those at below 18 degrees Celsius. Next, quick or fast freezing. It is the process by which the temperature of food is lowered to about 20 degrees Celsius within 30 minutes. Next, slow freezing. Here, it refers to a process where the desired temperature is achieved within 3 to 72 hours. Next, some foods are improperly wrapped through the freezer storage, which undergo freezer burn. Next, most bacteria require the water activity values above the 0.9 for their growth. In dried food that contain fat, oxygen, oxidative rancidity is a common form of chemical spoilage. Here comes about chemical preservative. Sodium benzoate was the first chemical preservative permitted in foods, preferably less than pH 3.6. Then sorbic acid. It works well below pH 6. Next, about cold sterilization. Destroy the microorganisms without rising the temperature. This process is known as cold sterilization. And next, UV light. It's a powerful bactericidal agent with the most effective wavelength of 2600 Armstrong. Next, about gamma rays. Electromagnetic radiation emitted from the excited nucleus of elements such as 60 cobalt and 137 cesium. Next, about sterilization, destruction of all microbes. Next, kefir. It is prepared by inoculating the cow, goat or sheep's milk with kefir grains. Next, humus. It's a fermented dairy product traditionally made from mare's milk. 0.7 and 2.5% alcohol. Next, Q fever. It is a disease caused by infection with Coccus burnetii. And next, Mycobacterium tuberculosis. It is a bacteria which causes tuberculosis in humans. Next, vacuum packaging. Here it stores the food in a vacuum environment, usually in an airtight bag or bottle. Then, salting or curing. It draws the moisture from the meat through a process of osmosis. And next about psychrophilic, microbe which can grow at refrigeration temperature or below low temperature. And 1 gray, it is equal to 100 rad. UV light has germicidal effect at wavelength between 252 to 80 nanometer. Then the irradiation dose range in kGY for inhibition of sprouting is 0.05 to 0.15. Next, the small ice crystals are formed due to fast freezing. Commercial and household refrigerators are operated at a temperature of 4.5 to 7 degrees Celsius. Here comes about the classification of stable or non-perishable food, semi-perishable and perishable food. So first, stable or non-perishable foods. So the examples are sugar, flour, dry beans, and semi-perishable foods, potatoes, and certain varieties of apples. Next about perishable food. Here comes meat, fish, poultry, most vegetables and fruits, milk. And next about rotting. It is a common form of bacterial spoilage in eggs. Green rot, Pseudomonas species, Pseudomonas fluorescens. Colorless rot by Pseudomonas and Acinetobacter. Black rot by Proteus and Pseudomonas aeromonas. Pink rot by Pseudomonas. Red rot by cereus. Custard rot by Proteus vulgaris. Proteus species and Pseudomonas graveolens also cause a condition in egg known as mustiness. Then fungi, Penicillium and Cladosporius species cause the spoilage of egg known as pin spots. And next about foodborne infection, example Salmonella. Then foodborne intoxication, example Botulinum toxin, Staphylococcal enterotoxin, Bacillus cereus toxin. Next, food toxico-infection. Here, combined form of food intoxication and infection. Example, Clostridium perfringens. Next, 4 pillars of good agricultural practices: economic viability, environmental sustainability, social acceptability, food safety and quality. And next, principles of hazard analysis and critical control point. Conduct hazard analysis, determine critical control point, establish critical limit, establishing monitoring procedure, establish corrective action, establish verification procedure, establish record keeping and documentation procedure. And next, HACCP steps. Assemble HACCP team, describe the product, identify intended use, construct flow diagram, on-site confirmation of flow diagram, conduct hazard analysis, determine critical control point, establish critical limit, establishing monitoring procedure, establish corrective action, establish verification procedure, establish record keeping and documentation procedure. And next, top yeast, Saccharomyces cerevisiae. For bottom yeast, Saccharomyces carlsbergensis. And blue cheese, Penicillium roqueforti. And next, Propionibacterium frutnertiae. It is for Swiss cheese to improve the flavor and assist the eye formation. And for cocoa fermentation, Candida krusei is used. For soya sauce and miso, Aspergillus oryzae is used. And for temp, Rhizophus oryzae. For citric acid production, Aspergillus niger is used, and for dextran production, Leuconostoc mesenteroids. Lyngbia majuscule causes Cigatura poisoning. Then, Scombroid fish poisoning has been associated with Proteus morganii and Klebsiella pneumonia. Travelers diarrhea caused by Escherichia. Next, typical operations and the food product. Blanching done in vegetable, churning in butter, conching chocolate, curing is done in meat, degumming in oil, malting beer, pearling sorghum, proofing bread, refining chocolate, rolling and withering process done in tea, smoking meat, winnowing cocoa. Then, important commercial products of yeast. Saccharomyces cerevisiae, the product beer, wine, bread, baker's yeast. So, the uses baking industry and brewing industry. Then, Saccharomyces roxy, the product obtained soy sauce and uses in food condiment. Then, Saccharomyces cerevisiae for ethanol production and uses fuel and solvent. Erythrithium ashby for riboflavin production uses vitamin supplement. Next, food containing aflatoxin. Aflatoxin produced by Aspergillus flavus and Aspergillus parasiticus in corn, peanut and cottonseed. Citrusin produced by Penicillium citrinum in wheat, barley and peanut. Ochratoxin produced by Aspergillus ochraceus in corn, barley, wheat and peanut. Patulin is produced by Aspergillus clavatus in silage and apple. Penicillic acid by Aspergillus clavatus in corn and beans. Alternarial by Alternaria tenuis in withered grain and sorghum. Cereulon by Fusarium roseum in corn, sorghum and wheat. Fumonicin by Fusarium by corn. Next, milk-borne diseases. Bacterial diseases. Brucella abortus, Corynebacterium diphtheria and Mycobacterium. Rickettsial diseases. Coxiella burnetii, this is Q fever. Then viral diseases. Enteroviruses, infectious hepatitis virus, tick-borne encephalitis virus, foot and mouth disease virus. So this is enteric fever, infectious hepatitis, tick-borne encephalitis, foot and mouth disease. Next, bacteria involved in food spoilage. Lactics by Lactobacillus, Leuconostoc, Pediococcus and Streptococcus. Then acetics, Acetobacter and Gluconobacter. Butyrics, Clostridium. Proteolytics, Bacillus, Pseudomonas, Clostridium and Proteus. Lipolyticus by Pseudomonas, Alkaligens, Serratia, Micrococcus, Bacillus and Clostridium. Saccharolytic by Bacillus and Clostridium. Pectolytic by Erwinia, Bacillus and Clostridium. Next, thermophiles. Bacillus, Clostridium, Lactobacillus and Thermophilus. Psychrophils, Alkaligens, Pseudomonas, Flavobacterium and Micrococcus. Halophils, Halobacterium, Sarcina, Micrococcus, Pseudomonas. Osmophils, Leuconostoc. Pigment formers, Flavobacterium, Serratia and Micrococcus. Slime or rope formers, Alkaligens, Enterobacter, Streptococcus, Lactobacillus. Gas formers, Leuconostoc, Lactobacillus, Proteus, Clostridium and Enterobacter. Then coliforms, Escherichia coli, Enterobacter aerogens. Then yeast involved in food spoilage. Saccharomyces in low sugar products. Zygosaccharomyces in honey, molasses, wine and soy sauce. Pichia in wines. Hansenella in beer. Deboromyces in cheese. Hanseniospora in fruit juices. Torlopsis in milk products, fruit juices and acid foods. And next, Rhodotorella in meat and sauerkraut. Candida in high acid foods, salt foods and butter. Britannomyces in beer and wines. Trichospora in beer. If you have any doubt, please ask on to comment section. Remaining units will be discussed on upcoming classes. So please subscribe our channel. Thank you.